Are we going? All right. Well, welcome to the first session of ASM Live at ICAC for the infectious disease meeting that's sponsored by ASM. We're in Chicago, and this is our first ASM Live session. I'm Jeffrey Fox of Microbe, the monthly magazine of ASM, and we will have a series of sessions over the next several days. We apologize for being late. We've had some technical difficulties of one sort or another. And a few ground rules. If those of you watching us over the internet, if you would like to ask questions, use hashtag ICAC. I won't tell you what ICAC means, but use those initials. And for those of you in the room, you're welcome to ask questions. Raise your hand, wait for someone to bring you a microphone, then please give us your name and institution. All right, so to the subject of our session. It's on antibiotic-resistant bacteria linked to seagulls in Miami Beach. And our guest is Patrice Nordmann of the, well, we'll say the Hospital de Bicetre in Le Kremlin, Bicetre in France. And Patrice, if you would begin by giving us a brief summary of the findings and maybe mention your collaborators, then we'll open up to questions. And thanks for joining.
Okay, many thanks for inviting me to try to make a summary of those findings. So it was a study designed mostly by our colleagues from the hospital, Jackson Memorial Hospital in Miami. And the group is led by Dr. Munoz-Price. And she had the idea, together with my colleague Laurent Poirel, to take some samples from the beach in Miami and to look those samples, those fishes from seagulls, who are spread on the beaches, and to look if those samples may have some multi-drug resistance bacteria. So she took, those samples, let's say 1 year ago in April 2010, 80 of those samples, then they grow those samples on specific media, and they study, the presence of the so-called extended spectrum beta-lactamase genes, which are multi-drug resistance bacteria in a way. And the findings were the following. Many of those samples, more or less 15%, they have the so-called ESBL genes together with other so-called plasmid-mediated cephalosporinase resistant genes. So the idea is that E. coli, which are in the faces of these seagulls, may be the source of multi-drug resistance bacteria. And to make a long story short, our opinion would be that these seagulls, they fly by definition. And so they take some water from where? From the garbage, from the surrounding of the cities, and they fly, and they have this in the mouth. And then it's quite logical to find those E. coli in the fishes. So they could be an important vector of spread of this multi-drug resistance bacteria.
Okay. So let's start with a little geographic discussion and directional discussion. I'm not quite sure. The seagulls, you think these resistant E. coli are making their way back into the general human population? And which way are things moving here? And does this account for what's going on in the human population in Miami Beach, in the hospitals in Miami, or is it more widespread, moving from one beach to another across the southern U.S.?
Clearly, this is difficult to know because E. coli is spread in the environment largely, in animals, and it is present in the gut of the human. So our idea is those E. coli are in the environment, they are taken by seagulls, and the seagulls may be a source of spreading of this ESBL. So the relationship in between infection in human and seagulls are difficult to be obtained clearly. It's difficult to know the real, the clear relationship in between infection in human with those multi-drug resistant bacteria and carriage of seagull with this bacteria.
And this phenomenon at this point is limited to Miami Beach just because that's where you looked. You haven't looked on other beaches at other species of birds, for example, because the seagulls are, yes, they're a garbage bird that moves around, but there are plenty of other waterfowl frequenting the beaches.
2 years ago, with people from Portugal, we have in our own group looked to the beaches in Porto, in Portugal, and we found exactly similar results. I mean, spread of ESBL genes, so those resistance bugs, in the faces of seagulls in Porto beaches.
I see. So
and another study has been published with people from Sweden reporting seagulls with these ESBL genes in the Riviera in France, near Marseille. So I think it could be a common fact that those birds, as other birds, may be important bugs that may spread these ESBL genes. But for flu, it has been reported that flu may be carried by birds.
Oh, yes. Now I guess we should pursue a little more precision about the ESBL genes themselves. Are they exactly the same as the ones that are found in clinical specimens, or is this a different sort of wild type ESBL that hasn't been found in clinical settings?
In the US, in 2011, the most important CTXM determinant is CTXM15. In those birds in Miami, they are CTXM15, so they're exactly the same.
I see. So, and are you, which direction is this, are these resistance factors moving predominantly? Are they going from the human population to the birds, or?
No, I think they are in the environment.
They're everywhere.
There is a reservoir of the CTXM15 which is well known, which is a cousin of E. coli, so-called Cluivera. So this bug is present in the environment, maybe in the plants, in the water. Then the gene is spread on a plasmid, then transferred in E. coli. And those E. coli are spread in the environment, in animal and in human. So I don't think that the bugs come from the human to the birds. I think in that cases, those birds have taken some water, contaminated water with E. coli. Those E. coli come from the environment or human? I don't know. But I think that those seagulls who are living in the garbage, they may have taken those genes inside themselves. So the clear relationship is difficult to establish, clearly.
Now, nobody is treating seagulls in Miami with antibiotics, right? So is this, and I say that facetiously, I don't assume we care about their health all that much. They're pretty tough birds. Is this, how to put the question, is this kind of neutral carriage, right? It's not being amplified. Is this likely to be a very important phenomenon, or is it going to be diluted out as the seagulls move to other areas where they're not replenishing their supply of the beta-lactamase genes.
No, I think the meaning of that could be the following. Those birds, they may fly for 300, 500, 1,000 kilometers with a little bit of water and foods. So the idea could be that they may spread in the environment to a large extent those resistance bugs. That's my understanding of the problem.
All right. So, for example, if there was a problem in the Miami area and the birds moved to North Carolina or New Orleans, then maybe they would be a means for distributing resistance factors that ordinarily wouldn't make their way to those distant places.
That's exactly my understanding of the problem.
Okay. And, but you have no empiric evidence to support this. This is a theory, a
right.
hypothesis.
No, I have no empiric, I have no experimental data to confirm what I'm saying.
And is the, you looked for one set of resistance factors, but it's quite possible that the birds are carrying other drug resistance factors, right? And
absolutely. I think those, you are absolutely right. We were just looking to some resistance to extended spectrum beta-lactam, but I think it's going to be exactly the same for the resistance of another big part of the antibiotic molecule, which are fluoroquinolones. I think resistance to those fluoroquinolones could be exactly the same as reported here in that study.
And are you planning some follow-up with different bird species, for example, or different areas? What's the distance? Because there are some very tantalizing facts here, but no solid answers, it seems.
We may, one of the future of this type of study is maybe to look to other antibiotic resistance determinants, and those days we are facing an emerging of multi-res and pan-drug resistance enterobacteriaceae. I mean E. coli or Klebsiella pneumoniae, which are resistant to almost everything, including carbapenem. So we may look to presence of this E. coli with resistance to carbapenem.
Okay, I see. And I was thinking, we happen to be in the Chicago area, and there are a lot of local geese that, as well as water birds. So this phenomenon might be widespread, working through other bird species, other geographic regions,
right. Right. I think this is a real problem of spread by birds of this antibiotic resistance determinant. I mentioned study performed Portugal, Marseille, and now I have in mind a study performed in Istanbul. And in Istanbul, in Turkey, they have identified plenty of pigeons with very similar, by the way, resistance determinants. So I think this is a worldwide phenomenon of spread of bacteria, and we are now in 2011, and those birds are taking what they can take, and in that case, they take water with E. coli, with ESBL, or similar cephalosporinase genes.
All right.
So I think this is a worldwide phenomenon.
Well,
that may, enhance the problem of multi-drug resistance determinant in community-acquired patients. Because what we are talking here is, I think, the consequence for the patient, not so much in the hospital, but for community-acquired patient. Because, in terms of infection due to E. coli, they are mostly located in community patients, not in hospital.
All right, I think we have some questions here in the audience. And again, once you have the mic, if you identify yourself and tell us who you write for or work with.
Dan Keller, Med School. Are you doing any epidemiology, or can you look at figures already gathered by CDC to see whether this has any clinical utility? Do people who go to the beach have more resistance genes in the bacteria they harbor, or do they get more diseases, or does this have no clinical meaning? And if you find that it is meaningful, what are you going to do about it? There's birds everywhere.
You are absolutely right. The clinical consequence of that in terms of epidemiological surveys could be, study performed in different area to see if there is some high or low level concentration of this multi-drug resistant bacteria. But you're right, we cannot kill all the birds and hope we are not kill all the birds all over our beaches. But you're absolutely right.
John Geaver, MedPage Today. So far you've only been talking about water birds. Is there any possibility that
well, he did mention pigeons. Which
okay, I was going to ask about pigeons and crows, which are much more widespread and numerous in inland areas. Is that also a concern?
Yes, I think it could be absolutely the same, right.
And also, birds in the environment would be, or at least garbage-eating birds, would be exposed to antibiotics. Penicillium molds are, ubiquitous in the environment, and is that potentially a factor in breeding resistance within the bird gut?
Yes, I think you're right. There is plenty of evidence that there is a lot of antibiotic residue in the environment. It has been looked, in sewage and in water coming out from the hospital in different environments. It is clear that in those cases there is plenty of antibiotic. There is a lot of chemicals that may resist to their spontaneous degradation, and in that case that may enhance the spread of this those multi-drug resistance E. coli. You are absolutely right.
Maren McKenna from Wired.com. So I want to ask for a little more detail about this question of whether the seagulls are vectors of multi-drug resistant bacteria to humans or sort of victims of exposure from human society. We just heard Dr. Livermore say downstairs that though CTXM15 is widely distributed across the globe, it's not always the same CTXM15. So do you have, or would it be possible to get molecular evidence of whether seagulls in Miami, for instance, are carrying the Miami clone, or the Miami plasmid, I guess, or whether they have transported something from France, from Sweden, from Istanbul, or somewhere else?
We didn't compare exactly the strain from the patient from the Miami area to those E. coli from the seagull from the Miami beach. But the genes are more or less the same, the so-called CTXM15. This is the most important CTXM determinant identified in the US. So this is not another type of CTXM. This is CTXM15, mostly reported in the US. So I think there is some kind of parallelism, at least in terms of gene. But the problem for doing this type of comparison is in E. coli, there is plenty of strains different in terms of E. coli. So I would imagine, I'm just saying that without having performed the study, but I imagine if we look to the patient from this Jackson Hospital, we look in E. coli and we may find maybe something like 200, 300, 400 different E. coli with the same ESBL gene inside. So there is a huge diversity of E. coli clone. So to make the relationship in between this huge diversity and the clone in seagulls, it's going to be tough. This is not like flu. In those cases, we have just one virus, and we can say, well, this is clearly the spread of just one single element. In that case, there is some overlapping in between the gene and the strains themselves that may carry those genes.
Laura Pullen, Clinical Podcast News. That sort of gets to my question. Have the studies been done with viruses or just bacteria?
Sorry.
Oh, you can't. Have the studies been done with viruses or just bacteria, the studies in wild birds?
No, what we have done here only with bacteria.
Okay.
Viruses, I mentioned, plenty of study reporting that birds are carrier of flu viruses.
Right.
And they have identified that in northern part of Canada. I remember a series of study reporting that.
Seagulls also?
Seagull, I'm not sure. No.
Okay.
Maybe other type of birds.
Do we have any questions from the internet? And we have exhausted the questions in the room. Do you have any summary you would like to say, or is there anything important about this study you haven't shared with us yet that you would like to say?
No, I think you have made everything. No, just my overall thinking of this study is the fact that seagull may travel for long distance, 200, 300, 500, 1,000 kilometers, and they may spread very quickly those resistance bugs within a given area. That, I think, the basics behind that.
All right, so just to think that we now know that we used to think about carrier pigeons, now we can think about carrier seagulls as a unique new bird species. We've been talking with Patrice Nordmann of Le Kremlin Bicetre in France. This ends our first session of ASM Live at ICAC. We have another session starting in, 15 minutes, I believe. So stay tuned. We'll be back with you again. Thanks very much.
Thank you.